Okay, good morning, everyone. Welcome back. Hope you're doing well, and hope you have some plans to have some fun this weekend. So, okay, please type yes into the chat window if you can hear me okay. Okay, great. All right, so just a couple things here. I noticed that a few of you, probably 4 or 5 of you, were able to successfully register on the ClassMarker website and take the practice quiz. So that's fantastic. Has there been any problems with ClassMarker so far? Okay, so I'll take your silence as though everything is okay. So please, for the rest of you, please be sure you have registered for ClassMarker uh, and taken the practice quiz by the start of next Tuesday's class, because I need to know that you're registered and that you feel comfortable with the website. So please do that this weekend. And if there's any problems, there'll be enough time that we can fix them before your first lecture exam this coming Thursday. A week from today, you have your first lecture exam. Okay, so to tell you a little bit about that, you have lecture exam 1 coming up a week from Thursday, from today. And what you can expect this afternoon in this afternoon's email, in addition to the normal information that I give you, I will also send you an exam study guide. That exam study guide will have information which is suggestive of content that you need to master, that you need to understand in order to do well on the exam. For example, there's a section of the study guide where I list specific vocabulary terms that you need to know to do well. Now, I don't tell you all the vocab. I tell you about 50% of the vocab, okay? So there's going to be about 34 questions on this first exam that are specifically designed to test your understanding of the new vocab in the chapters that we're currently covering. So there'll be 34 questions testing vocab. And on the study guide, I give you 17 of those vocab terms. So I give you 50% of the vocab as a way to get your intellectual juices flowing. Okay. Now the terms on the study guide, they will either be the answer to a question or they will be a topic that you need to master in order to answer my question. For example, one of the vocab terms is pH. You know, we did talk about pH. So pH might be the answer to a question on your exam, or the concepts surrounding the topic of pH, such as hydrogen ions, such as acids, such as bases. Any of those things could also be tested on vocab-related questions. Okay, so once again, the vocab could be the answer to a question, or it will point you in the direction of the topic you need to understand in order to answer a question. Okay, so, you know, let's be honest, at this time with our remote learning scenario, you folks do have it a little bit easier than you would if you were in class together. And that's because you get to take your exams with your textbook and notebook sitting in front of you. So we are trading a little bit of educational rigor for public safety at this time. And we have to admit that, okay? Because, you know, if you were taking the exam in class, at least for the essay portion of the exam, you would not have your textbook and notebook with you. But you will have that at home as we take the class remotely this summer, okay? But you still need to prepare, folks. You still need to study. You still need to memorize, because you will not have enough time to look every answer up, okay? So please don't rely on the strategy that you can look up the definition of 34 vocab terms and expect to do well. That will not work. Okay, you have to prepare for the exam. Okay. So the study guide is going to have a list of vocab terms, and it's also going to have several of your essays that you will be tested on next Thursday. You know, I give you several of the essays up front because I intentionally want you to go out and research the topic and prepare a good answer ahead of time, because I want a good answer on your exam. So I give many of the essays ahead of time, but there will still be a couple essays that I don't give you the exact essay. I simply tell you the general topic. For example, one of the major essays on your exam is going to be osmosis. I'm not going to tell you specifically what that's about, but I'm telling you, you need to prepare to explain osmosis and apply your knowledge of osmosis in order to answer the exam question next Thursday. Okay, so the study guide will push you in certain directions to help you know how to study, but it's not going to give you everything, because part of your responsibility is to study, to think, and to learn. Do you have any questions at this time?
Did you say the test will be start next Thursday?
Next Thursday, that's correct. It will open up at 7 a.m. and it will close at midnight. Okay, so you have
not until 9:30 of the day, only at Thursday and then Thursday, right?
That's correct. You may only take it next Thursday, a week from today, between 7 a.m. and midnight. Okay.
Okay.
All right. Okay, so please register for class marker. Some of you did register for it, but you did not take the practice exam. Please log back in and take that practice exam. And I'll ask you folks on Tuesday after the weekend if you need help with anything. Okay? All right. So what we're going to do today is fairly quickly, maybe in about 20 minutes or so, wrap up chapter 3, and then we're going to be on to chapter 4. Okay? So here we go. I'm going to start sharing my screen as I usually do and go to a Microsoft Word document. Okay. So at this moment, you should see the Microsoft Word document in the chat window along the side, so you can get a hold of me if you need me. Okay, so folks, is it correct that the last topic we covered on Tuesday was osmosis? Is that the last thing we did? Please let me know. Okay, thank you, Matt and Catherine. Thank you, everyone. All right, so we are talking about methods of moving cargo through the plasma membrane. And remember from your notes last time, I had a table where I had passive transport mechanisms and active transport mechanisms. And so last time we covered all the passive ones: simple diffusion, facilitated diffusion, and osmosis. So today we're going to take a few minutes and talk about some of the active transport processes, okay? And we won't spend too much time on this, but I do want to be sure that it is in your notes. So, all right, so the first active transport process is active transport, okay? Active transport is the name of an active transport process, okay? So a couple of things here. This is active transport is basically facilitated diffusion in reverse. Okay. Okay, and so what I mean by that is, let me give you the big long English definition. Okay, remember our definition for facilitated diffusion was the movement of a solute across the membrane with the help of a carrier protein from an area of high concentration to an area of lower concentration. Well, we're basically the same thing here, except rather than moving the solute from high to low, we're going to move it from low to high. Okay. Okay, so I will emphasize that in your notes. We're going to move the solute from an area of low concentration to an area of higher concentration. It's exactly the opposite direction that the solute would move for facilitated diffusion. Okay, and because of that, because this is sort of against the grain from a thermodynamic perspective, we actually have, the cell actually has to consume energy to move the cargo. You know, as a silly example, imagine that you're at the top of a flight of stairs, and as you're standing at the top of that flight of stairs, if, you know, don't do this, but if you were to let your leg muscles go weak and you were to collapse, you would simply fall down the flight of stairs because gravity would pull you down. And you would fall down a flight of stairs without using any of your own energy in the muscles of your legs or anything, because you don't need to use any energy to fall down a flight of stairs to go from high to low. But if you were at the bottom of your stairs and you wanted to crawl back up to the top of the flight of stairs and to go against gravity, you would have to burn energy inside your leg muscles to lift your body from the low position to the high position up the flight of stairs. So anytime you want to move cargo, anytime you want to move solute from low to high concentration, the cell actually has to consume energy. And the type of energy we're talking about, and which we kind of know at the beginning level, is ATP, okay? So this will require cells to consume ATP to move cargo against the grain, so to speak. Okay. All right. So you might think that, I mean, obviously we do that every day, you know, because we're walking around and we climb flights of stairs. That's just one practical example of where energy is going to be used. But you also have to appreciate that active transport is also used constantly, every moment of every second in your nervous system to maintain the proper voltage across the plasma membrane of your neurons in your brain that allow you to be awake, that allow you to be conscious, that allow you to be alive. Okay, so active transport, as you just sit there on your chair at the house, and even if you try to clear your mind and not think of anything particularly intense, your brain is still consuming ATP to support the active transport process to maintain the healthy voltage across the plasma membrane of your neurons. So you are doing it constantly. Even when you're asleep, you are doing it constantly. Do you have any questions? Okay, not hearing anything, so I'm going to keep rolling. Okay, the two remaining active transport processes that I simply want to get into your notes, but they're not too big of a deal for us right now, are the endocytosis and exocytosis. Okay, endocytosis is when the cell wraps its own plasma membrane around some kind of big cargo that the cell wants to bring inside. Quinn, yes, dreaming takes energy. It absolutely takes biochemical energy. That is correct. In fact, you know, brain imaging studies very clearly show that your brain is metabolizing and using more energy during periods of dreaming, okay, during REM sleep. Okay, so endocytosis is a process of internalizing. By that, I simply mean bringing inside the cell, process of internalizing large cargo by wrapping the plasma membrane around the cargo. Okay, so last time we mentioned that simple diffusion would move cargo that's very tiny and electrically neutral, such as oxygen molecules and carbon dioxide molecules. And we also said last time that facilitated diffusion could be used to move larger cargo like glucose. And then we switched gears and started talking about osmosis, where we said, well, now we're talking about the movement of the solvent, not the solute. So let me mention up here underneath active transport, an example of the type of cargo would again be, okay, cargo that could be moved by active transport include glucose. So again, the same size cargo that I could move by facilitated diffusion, if I want to move that cargo in the opposite direction from low to high, well, then I could use active transport to move that cargo. So cargo that could be moved by active transport would include glucose, and it would also include things like, you know, hydrogen ions or sodium ions or potassium ions, things that have a charge on them. If I need to move that charged material from an area where it's low in concentration to an area and high in concentration, I can use active transport to achieve that goal. Now, for endocytosis, the example of cargo that I would use here would be an entire bacterium, okay? In other words, sometimes cells will intentionally bring in bacterium inside themselves as a way to destroy the bacterium and protect the body from greater infection, okay? So that process of bringing in a bacterium would be an example of endocytosis. And obviously, a bacterium is made of billions of molecules. So the cargo, the size of the cargo is dramatically larger when we're talking about the endocytosis, okay? So the entire bacterium, which is huge cargo. Okay, your book has a picture of that on page 50 of your textbook, figure 311. Okay. And in just a moment, I'm going to show you a quick little video that gives you some visualization on this. Okay. So endocytosis is one mechanism of active transport for moving very, very large cargo into a cell. And if we move the cargo in the opposite direction, that would be exocytosis. The process of removing large cargo from the cell by pushing it through the plasma membrane. Okay. An example here of cargo that might be done this way are tiny little bubbles that are made of lipids that contain molecules of RNA in it that can then leave the cell, float into your bloodstream, and be absorbed back into another cell at a distant location. For example, and then, okay, let me write that down, then I'll give you a more practical example. Cells sometimes export by exocytosis lipid bubbles containing a variety of molecules, including RNA, okay? This cargo can leave the cell. I see your question, Quinn. This cargo can leave the cell, float in the bloodstream to a distant location where a second cell can take the lipid bubble inside and use the information in the bubble to change its behavior. Okay. Now, an example of my example. Quinn, don't cells get rid of waste this way too? Yes, they sometimes do that, Quinn. Okay. An example of my example, which is kind of has a negative connotation, would be cancer. When cancer cells metastasize, meaning they spread, they spread from their original location. Sometimes what is really happening is a cancer cell at one location of the body releases this lipid bubble through exocytosis. And it has this lipid bubble from the cancer cell has instructions so that when another cell at a different location takes in this message from the distant cancer cell, it changes the behavior of the second cell in such a way that that second cell starts to become cancerous. So you can see that the cancer literally spreads that way by converting cells from the healthy state to the cancerous state. Sometimes what is really happening is cancer cells are reprogramming healthy cells at another location to become cancerous through instructions in the lipid bubbles. Okay. This is an active area of research. It's fairly contemporary. I mean, scientific community has probably been aware of these lipid bubbles and their relationship to cancer for maybe 20 years. So there's still a lot we don't understand, and current therapies to deal with these things are not so good yet. So there's a lot of work to be done. Okay. Is it possible to create a cell to reprogram cancer cells to not become cancer cells? In theory, that should be possible, Pete. But in theory, I could grow wings and fly. So in theory, yes, but making that a practical reality has been very elusive. But yes, we think it should be possible, but we don't really have the technology to do that yet. Okay, any other questions? Okay, so gang, rather than draw this out any longer, what I'm going to do is show you a quick little video that summarizes endocytosis and exocytosis so that you can see this. Okay, here we go. Bear with me a second while I bring up the endocytosis and exocytosis video. Okay, so you should at this point see YouTube, and I'm just going to let it play out for itself. Here we go.
The substances taken in by single-celled organisms are often particles or large polar molecules that cannot cross the hydrophobic plasma membrane. Many single-celled eukaryotes employ endocytosis to ingest such food particles. In this process, the plasma membrane extends outward and surrounds the food particle. Cells use three major types of endocytosis: phagocytosis, pinocytosis, and receptor-mediated endocytosis. If the material the cell takes in is particulate, such as a bacterium or a fragment of organic matter, the process is called phagocytosis. If the material the cell takes in is liquid, it is called pinocytosis. Specific molecules, such as low-density lipoproteins, LDL, are often transported into eukaryotic cells through receptor-mediated endocytosis. Molecules to be transported first bind to specific receptors on the plasma membrane. The interior portion of the receptor protein is embedded in the membrane. The protein clathrin coats the inside of the membrane in the area of the pit. When an appropriate collection of molecules gathers in the coated pit, the pit deepens and seals off to form a coated vesicle which carries the molecules into the cell. Exocytosis is the reverse of endocytosis. This process results in the discharge of material from vesicles at the cell surface to the outside of the cell.
Okay, gang. So there you saw a quick little visualization of some of the major ideas we've been talking about. Obviously, there are subtypes of endocytosis, which the video referred to as, phagocytosis and pinocytosis and receptor-mediated endocytosis. Okay, those go a little bit further than we would probably go in our course, but nonetheless, you've seen them. Okay, so at this point, do you have any questions about any of the active transport processes? Okay, not hearing anything. I want to keep rolling. So the next and final topic of chapter 3 that I at least need to cover a little bit with you is aerobic cellular respiration. We started chapter 2 with that chemical summary equation of aerobic cellular respiration, and you did really well with that. Folks, we're not going to go into the biochemistry of the process. That would be for a more advanced course, but I still need to give you an overview of the kind of things that we're thinking about for cell respiration, and these things are in your book. The conversation starts on page 57, goes to page 58, 59, 60, and 61. So there's a considerable amount here, and I'm going to try to summarize some of this for you. Okay, and aerobic respiration, aerobic cell respiration overview. Okay. So just remember some things, right? Remember that aerobic cell respiration refers to all of the chemistry, and it's fairly complicated chemistry, but we're not going to do that. All the chemistry involved in extracting energy from the food that you eat. All right, and honestly, your book doesn't really give you all the chemical detail. It hides a lot of it from you, and that's okay because this is just a beginner level course. But on page 58 of your textbook, figure 322, there you have glycolysis in a very cartoon fashion, and that's fine. But there are some steps there that are intentionally not being shown to you, okay? Same thing for page 59, figures 323 and figure 324, there are also some additional steps that are not shown to you. And on page 60, figure 325, some additional steps that are not shown to you. Okay, so don't worry about what's not being shown. I mean, if you happen to be interested in that, you know, I can point you in the right direction to get some more detailed information. But at any rate, okay, aerobic cell respiration has 4 phases, but only 3 of those phases produce energy. In other words, only 3 of those phases produce ATP. Okay. So let's take a look at that. Look at this. Okay. Phases of aerobic cellular respiration. Okay. The first phase is glycolysis. Okay. I am going to show you a video on this so that maybe you get a little bit of more comfort level with what we're talking about. So if all this stuff seems very distant or confusing because you really haven't covered this stuff in the past, you know, don't give up, keep at it, and there's going to be some more visual information coming your way in just a minute or two. So glycolysis, which you see on page 58, okay, glycolysis refers to a group of chemical reactions that take place in the cytoplasm of the cell. It's a group of chemical reactions that take place in the cytoplasm of the cell. You don't have to know what those reactions are. You don't have to memorize them. You don't have to balance the chemical equation. You don't even have to write them out. Okay, that's not, you know, that would be for our biology major students, and you are not that if you're taking this course. Okay, so glycolysis refers to a group of chemical reactions that take place in the cytoplasm. And what I would want you to know is that glycolysis results in the production of 2 net ATP per 1 glucose molecule broken down. And obviously the glucose is coming from your food. Okay, the glucose is coming from your food. Okay, so question coming through the chat window here. Well, I'll put it in your Word document. Folks, what does the term net mean? What does the term net mean in this conversation? Okay, Quinn, you say total gain. I'm not sure what you mean by total gain. So for example, so Quinn, can you look at, do you have access to your textbook right now? Okay, so Quinn, take a look at figure 322. And I'm going to ask you a few questions. Okay, and everybody please try to follow along here, and maybe we'll need your help. Maybe Quinn will need your help. Okay, so Quinn, how many ATP are manufactured according to the information in figure 322?
4 are apparently made,
Okay.
but 2 are used.
That's exactly right. So is that what you meant by total gain?
Yeah, that's what I tried to mean, but I'm not very good with words.
No, that's okay. That's okay. I just needed to clarify. So you did have the right thinking. I just couldn't tell from some of the words. Okay, does everybody see that? That if you look at figure 322, okay, the cell consumes 2 ATP to manufacture 4 ATP. So that means you manufacture 4 ATP, but you had to use 2 ATP to manufacture the 4. So that means the excess ATP, the profit of ATP that you get, the net ATP that you get is only 2, okay? So the profit or net ATP or excess, profit net excess ATP is 2, okay? All right, so does that make sense, folks? Let me get some feedback from everybody online. Do you understand what net means? Okay. If you do not understand what net means, please speak up so we can spend more time on this. All right. Okay, please let me know. You know, there are no dumb questions, and there is no shame in saying I don't understand. There is no shame in that. So please speak up when you need help. Okay. So, all right. So glycolysis does that. Glycolysis is also known as the Embden-Meyerhof pathway, after doctors Embden and Meyerhof that helped characterize the process. Okay, so right now, folks, that's basically all I would need or want you to know, okay? I mean, if you want to go further in your study time, go for it. I encourage you to do that, okay? All right. So the next part of aerobic cellular respiration is the transition reaction. Okay, you see that there on page 59. The transition reaction does not make energy. Okay, no ATP is made. The purpose of the transition reaction is to take the molecules that are made at the end of glycolysis and push them into the mitochondria so that the next step of the reaction can happen in the mitochondria, okay? So let's put that in. Okay, so the transition reaction is very important because if it didn't work properly, most of the energy from aerobic cellular respiration could not be made because the material to make it would not be in the right location. That being said, while it is very important, again, it itself does not make any energy. Okay. All right. The next step is the citric acid cycle of page 59. Okay. This is also known as the Krebs cycle or the tricarboxylic acid cycle, right? It's known by 2 other names, okay? And let's see here. I will point out a couple things. Let me go back up to the transition reaction. I will point out, well, I'm going to ask you folks. Okay, I'm going to ask you folks. Okay, Fiona. Fiona, do you have access to your textbook right now?
Yes.
Okay, Fiona. Fiona, when you look at figure 323 on page 59, what gas that we typically exhale from our lungs is actually made during the transition reaction? Once again, what gas that we normally exhale from our lungs is made during the transition reaction on page 59? Do you see it?
Page 59.
Figure 323. What molecule there is a gas that we exhale from our lungs?
Uh,
So Fiona, let me help you out here. Normally when we inhale, we take in oxygen, right?
yeah.
So when we exhale, what do we exhale?
The CO, carbon dioxide.
Very good, CO2, and that's what I'm pointing out. Some of the CO2 that we exhale is actually made by cells during the transition reaction.
Mhm.
So thank you, Fiona. Some CO2 is made here. I'll also point that out with the citric acid cycle. Citric acid cycle makes additional CO2 that we exhale from our bodies. Okay, and in terms of an energy tally, we manufacture an additional, okay, so before I tell you that, right, so the transition reaction has taken the end product of glycolysis, which happens to be pyruvate, okay, and the transition reaction moves it into the interior of the mitochondria. So the citric acid cycle takes place inside that mitochondrion. Okay. And when the chemistry of the citric acid cycle is done doing what it needs to do, we end up with an additional net 2 ATP. Okay, so we manufacture an additional 2 net ATP per glucose. Okay. And then finally, the last stage, the last phase, if you will, of aerobic cellular respiration is in fact going to be the electron transport chain. And this takes place in the mitochondrion, but I can be even a little bit more precise here. The electron transport chain literally happens across the inner mitochondrial membrane. If you look carefully at the mitochondrion on page 60 or page 61, you can see that the mitochondrion actually has two membranes, an outer membrane and an inner membrane. Well, the chemistry of the electron transport process actually happens across the inner mitochondrial membrane. All right. And so what I would want you to know about this is the amount of energy that's made during the electron transport chain process. By electron transport, the cell will manufacture an additional 32 net ATP per glucose. Okay, so our grand tally for ATP production by aerobic cellular respiration is what? What's our grand tally? For every molecule of glucose, how many molecules of net ATP do we manufacture? Okay, thank you, Matt. It is 36. Okay, all right. All right, folks. How quickly does the whole process happen? We're talking well, with the enzymes of the cell being as fast as they are, you could conceivably manufacture, because there's more than one enzyme working at the same time to do this process, Quinn. Literally, if a cell is so motivated to do it, you could manufacture, I would say, probably 1 million ATP per second. It's definitely way more than 1, 2, or 3. Okay. All right. Other questions? Okay, so let's take a look at a video which is going to give you some, a cartoonish overview of this process. It might still seem a little bit foreign to you. So let's watch this video. It is a little bit long, folks, and I'm going to ask you to please stay focused, right? We're showing this, we're using class time because we want you to understand. So try to stay focused as you watch this. I will send you a link to it later so that you can watch it again. Here we go.
Turn that bite of food into a chemical that a cell can recognize and use as energy. The first step is altering the food into its component chemical compounds and then getting those molecules into your cells. That process is called digestion. Once inside your cells, the process of turning that bite of food into useful energy by cellular respiration begins. The process of digestion results with carbohydrates and other molecules being removed from the consumed food and transported into the bloodstream. From there, nutrients, like the carbohydrate glucose, will leave the bloodstream through a capillary wall and enter a tissue cell. Once inside the cell, cellular respiration will completely oxidize the glucose molecule, releasing high-energy electrons. The overall goal is to make ATP, a storage form of energy for most cells. Cellular respiration is a 4-stage process that begins with glycolysis. Glycolysis literally means splitting sugars, and it is the first step of cellular respiration, occurring in the cytoplasm of the cell. Glycolysis consists of 2 distinct phases, an energy investment phase and an energy harvesting phase. In the energy investment phase, 2 ATP molecules transfer energy to the glucose molecule, forming a 6-carbon sugar diphosphate molecule. This molecule splits, and the energy harvesting phase begins. During this phase, the 2 3-carbon molecules are converted to pyruvate, and ATP is formed. Glycolysis is a 10-step reaction that involves the activity of multiple enzymes and enzyme assistance. In the process, a net of 2 molecules of ATP, 2 molecules of pyruvate, and 2 high-energy electron-carrying molecules of NADH are produced. When oxygen is present, the pyruvate molecules and NADH enter the mitochondria, and the next stage of cellular respiration begins. The next stage of cellular respiration involves the movement of pyruvate into the mitochondria, where it undergoes oxidation. Each pyruvate molecule is converted into a compound called acetyl-CoA. In the process of pyruvate oxidation, electrons are transferred to NAD, producing NADH, and a carbon is lost, forming carbon dioxide. The next stage is the citric acid cycle, also called the Krebs cycle. Here, acetyl-CoA will bind with a starting compound called oxaloacetate, and through a series of enzymatic redox reactions, all carbons, hydrogens, and oxygens in pyruvate ultimately end up as carbon dioxide and water. The pathway is called a cycle because oxaloacetate is the starting and ending compound of the pathway. For every glucose that enters glycolysis, the cycle completes twice, once for each molecule of pyruvate that entered the mitochondria. During pyruvate oxidation and the citric acid cycle, a net of 8 NADH, 2 FADH2, 2 ATP, and 6 CO2 are produced for each glucose molecule. In order to understand how the majority of the energy is produced by aerobic respiration, we need to follow the NADH and FADH2 molecules to the next stage, the electron transport chain. The electron transport chain is a series of membrane-bound carriers in the mitochondria that pass electrons from one to another. As the electrons are transferred between the membrane proteins, the cell is able to capture energy and use it to produce ATP molecules. Proteins in the chain pump hydrogen ions across a membrane. When the hydrogen ions flow back across the membrane through an ATP synthase complex, ATP is synthesized by the enzyme ATP synthase. Oxygen acts as the terminal electron acceptor. By accepting electrons, oxygen is reduced to form water, a byproduct of the electron transport chain. All the high-energy electron carriers from the previous stages of cellular respiration bring their electrons into the chain. From this, the bulk of ATP from the entirety of cellular respiration is produced, a net of 32 to 36 ATP. In summary, we have seen how the 4 stages of cellular respiration are responsible for converting the energy found in the glucose molecule into ATP, the energy battery of the cell. On average, 36 ATP molecules are produced per glucose molecule that entered the cell. In the process of producing ATP, oxygen is brought in from the bloodstream to be the final electron acceptor in the electron transport chain, and the carbon dioxide that is produced as a byproduct is released. The goal of cellular respiration is to transfer the energy from the food that we eat daily into ATP that our bodies can use.
Okay, folks, so a little bit of a long-winded explanation there, but do you have any questions right now? You know, this whole process, all of this chemistry, we could take a month to go over in detail, and that's really not the focus of our course. So we're just covering it from a surface level in a summary fashion. Do you have any questions you want to ask about it? Any questions about the video? Please raise your hand if you are grateful that you're not a biochemist. Okay, very good. Okay, that's what most people think, and that's okay. That's totally okay. But hopefully you would agree that we at least need some people that attempt to understand this, because when this process goes wrong, we get processes like cancer happening in the body. Okay, so one final little thing, and then we will wrap up chapter 3. Okay, the last thing I need to mention to you is an alternative to aerobic cellular respiration that can sometimes happen when your cells are experiencing an oxygen debt. Do you know what I'm talking about? If your cells are experiencing a lack of oxygen, they cannot perform aerobic cellular respiration. In such a case, what will your cells perform? Matt, do you have a lack of oxygen? Okay. Okay, very good. Fermentation, that's right, Quinn. Okay, so fermentation is mentioned on page 60 of your book. Okay, so an alternative to aerobic cellular respiration is fermentation. Okay. Now, I'm not going to go into that because fermentation, I'm sure you've heard that word before, and why we use fermentation you know, society uses fermentation all the time to make food products, right? There's fermentation to make cheese, fermentation to make bread, fermentation to make wine and general beer and other alcohols, right? Fermentation is a broad topic that you could have a college course in, so we're not going to touch that. But in contrast to our current topic, our current conversation, okay, if your cells are experiencing a lack of oxygen, the cells will switch from performing aerobic cellular respiration to fermentation. Okay? Well, you might say, well, under what circumstance am I going to have a lack of oxygen? Well, just think about the last time you exercised heavily and you started to feel winded, right? If you exercise heavily and you start to feel winded, right, you can't bring in the air fast enough to power your muscles. So in such a case, if that condition goes on long enough, the cells of your body will switch to a fermentation chemistry. Now, the big deal about this is, excuse me, if your cells perform fermentation, okay, then you only get 2 net ATP per glucose. Okay, that's all you get, folks, right? Our grand tally for aerobic cellular respiration is 36 net ATP, but for fermentation, you only get 2 net ATP. So fermentation is very much inefficient compared to aerobic cellular respiration. You cannot get as much usable energy from fermentation chemistry as you can from aerobic cell respiration chemistry. Okay, folks, do you have any questions? So Emily, you asked, how would ATP enter other cells, for example, as a muscle cell in order for it to contract? Emily, generally speaking, ATP does not cross the plasma membrane into other cells, but each individual cell of your body has the ability to make its own ATP, right? So the muscle cell would make its own ATP. It would not export the ATP to another cell. Does that make sense? Okay, there are no dumb questions. It's a good questions. Keep asking. Okay. So folks, I'm going to switch gears now and I'm going to start chapter 4. Okay? Chapter 4 is our introduction to the major tissues of the body. Okay, and it's easy for me to talk about them in a fairly short period of time, but it's not necessarily easy for you to read about them and understand them in a short period of time. Okay, so let's continue here. Chapter 4, tissues. Okay. The title of chapter 4 is Body Organization and Homeostasis. That's basically some ideas we're going to talk about. But an emphasis of chapter 4 is, in fact, introducing the variety of tissues to you that make up your body. Okay, so let's remember some things. Okay, maybe some things that I've said, or maybe I didn't say it, so we'll write it down at this point. So remember, number 1, a cell is the basic unit of life on the planet. Number 2, a tissue is a group of cells that stay associated with each other to perform a common function. Okay, that common function might be protection. Okay, might be protection. Number 3, an organ is a three-dimensional object made of multiple different tissues that stay associated for a common function. And so, you know, that common, you know, an organ like your heart, right? It's made of multiple different kinds of tissue, right? Your heart, obviously, when you think of the heart, you probably think of cardiac muscle tissue, right? But there's still, you know, a lot of nervous system tissue embedded in the fabric of the heart. And obviously the nervous system tissue plays a huge role in helping regulate your heart rhythm. Okay, so there's lots of factors playing into it. So we're not going to emphasize our knowledge of cells in this chapter. We're not going to emphasize our knowledge of organs. We're going to emphasize our knowledge or basic knowledge of tissues. Okay. In your body, there are 4 major tissue types, and I think we did list this down already. They are epithelial, connective, muscle, and nervous. Okay, these are the 4 broad major categories of tissues that are made up of cells in which themselves interact with each other to form organs to do what your body needs achieved. Okay, so let's start with epithelial tissue. And all we're going to do is walk through this a little bit and summarize a few things. You're going to have to spend some time thinking about these things. And honestly, you know, one of the labs we would normally do would be a lab where you look at the tissues under the microscope and you get comfortable with these things. And that's going to be a little bit harder to achieve since we're all working at home at this point. All right, so epithelial tissue here. Okay. Epithelial tissue is made of closely packed cells that form a continuous layer in the body, either in the body or on the surface of the body. Okay. Epithelial tissue is made of tightly packed cells that form a continuous layer or surface. This surface can be internal or external, right? I mean, when we think epithelial tissue, probably it's probably fair to say that the average person probably thinks about their skin. And the skin is a good example, right? The surface of your skin is made of tightly packed cells called the epithelial tissue. That's very true. But please understand that epithelial tissue does not necessarily have to be on the surface of your body. It can actually be inside your body as well. So because of that, there's 2 broad types of epithelial tissue. Okay. And they are membranous epithelium. Okay, membranous epithelium. Its function generally is to protect, secrete, and absorb material. So protect your body. Think about how your skin provides a physical barrier to protect the inside of your body from microorganisms like bacteria and viruses. Also think about how sometimes when you overheat in the summertime, you sweat, right? You secrete material, you sweat in order to lower your body temperature. And then also think about in the wintertime when your skin gets dry and chapped because maybe you're washing it too much or whatever, that maybe you put on some skin lotion and that lotion gets absorbed into the surface of your skin, the surface of the epithelium. So membranous epithelium, its job is to protect, secrete, and absorb material. Okay, and we can have membranous epithelium that's external, such the outer surface of your skin. And you can also have membranous epithelium, which is internal. And, you know, there's lots of things we could say there, but let me, you know, think about it. The lining of the inside hollow space of your urinary bladder where your urine is stored, well, that lining is made of membranous epithelium. Similarly, your blood vessels. If we think of your blood vessels as tiny little garden hoses, right? You think about your garden hose at your house, right? You hook it up to a faucet and you run water through the hose. Well, that water is running across the internal inside surface of the garden hose. Well, blood vessels have blood that run across that internal surface. Okay, that internal surface of a blood vessel is made of membranous epithelium. Okay, such as the lining of blood vessels and hollow organs like the urinary bladder. Okay. All right. So, you see on page 67 of your textbook, figure 4.1, there's a whole bunch of different subtypes of epithelium. And folks, this would be a topic for our lab next week, not today. Today is about the microscope, but our lab next week would be, in fact, about these different kinds of epithelium and different kinds of subtypes of tissues that exist. So we're going to cover some of that kind of ideas, some of those ideas next week in our lab time together. So I'm not going to do that with you here. Let me say this. For your very first lecture exam, a week from today, I will not have any pictures of tissue that you have to identify. Okay, for your lecture exam in a week, I will not have any tissue pictures that you have to identify. But for your first lab exam, I will have pictures of tissues that you have to identify. Okay, so please know that that's going to be part of your beginner skill set that we're going to start to develop here. Okay, do you have any questions? Okay, so we're talking about epithelial tissue. I said there's two broad types. The first was the membranous epithelium, and we said that there can be external and internal varieties of that. Okay. Pete, you asked a question, how many questions will be on the exam? On your lecture exam next week, there are going to be 34 vocab-related questions and about 8 essay questions. Okay, so the second type, the second broad type of epithelium is glandular epithelium. Okay. Okay. Glandular epithelium is that epithelium that forms the secretory part of glands. Okay. And so I bet you in your background, probably in health class in high school, you know, you had a conversation about exocrine glands and endocrine glands, right? Exocrine and endocrine glands. So we have exocrine glands and we have endocrine glands. Okay. And I'm going to come up and make some statements about them. But before I make any statements, here's a question to you. What is the major difference between exocrine glands and endocrine glands. What is the major difference, folks? Does anybody know? What's the major difference between exocrine and endocrine? Okay. Well, okay, Alyssa, thank you for answering. You are not incorrect. There are structural differences in terms of the ducts. That is not incorrect. But that's still missing the big picture here. Okay, it seems like maybe I've got you on this one. Folks, the major difference is this. Okay, Quinn, you say one is internal and one is external, and that's a very common misunderstanding because that is not true. Okay? The major difference is that the material secreted by exocrine glands never accesses the bloodstream, but material secreted by endocrine glands does enter the bloodstream. It has nothing to do with internal or external. Okay? Substances secreted by exocrine glands do not enter the bloodstream. Okay? While substances secreted by endocrine glands do enter the bloodstream. Okay? All right. I mean, in a very simplistic way, think about, think about the sweat glands under your armpit, right? Obviously, when you produce sweat, obviously the sweat is on the surface of the body, so that's external, and that's why people often think about it this way. But it's more appropriate to think about it as sweat as something that is never getting into your bloodstream. In a similar fashion, endocrine tissue, for example, your pancreas, which manufactures and releases insulin to control blood sugar. Well, obviously that insulin does have to enter your bloodstream to regulate sugar levels in the body. Okay? So exocrine, whatever it releases and wherever it releases it, it doesn't enter the bloodstream. Endocrine, it does enter the bloodstream. Okay. Okay. So yeah, for exocrine glands, examples, could be, okay, so exocrine glands examples could be your sweat glands, could be your salivary glands in your mouth, right? Those are obviously at the surface of your body, they're external, but then you also have glands in your stomach, inside your body, in your stomach, inside your body that produce acids so you can digest your food. And that acid never gets into your bloodstream, right? So there's an example of an internal exocrine gland system. Okay. All right, then endocrine glands. Well, endocrine glands could be examples. Examples could be the pancreas that releases insulin. It could be your heart, right? Your heart, believe it or not, there is glandular epithelium in your heart. And one of the major hormones that tissue manufactures on behalf of your heart function is called atrial natriuretic peptide. And we'll get to that at another section of our class. But just so you know, atrial natriuretic peptide is a hormone made by the glandular epithelium of your heart, which then gets into the bloodstream, floats down to your kidneys, and tells your kidneys, hey, start making more urine. Okay? Okay. All right, folks. So this is just a gentle overview of epithelial tissue. Do you have any questions right now? Okay, I hope you're still conscious. You've not passed out at home. And so let's continue. The second kind of tissue we need to talk about is going to be the connective tissue. Connective tissue, okay. Connective tissue is kind of unique among the tissues because you know at this point that tissues are made of cells, groups of cells. We put that into your notes. But connective tissue is unique because not only is it made of cells, but it's also made of an extracellular glue that holds the cells together very tightly, and the proper term for this substance is called matrix, the extracellular matrix. So connective tissue is the only tissue that has cells and an extracellular matrix. Okay? You might ask yourself, well then, how do other tissues have cells that stay connected? Well, if you've read through chapter 4, you probably know the answer. But if you haven't yet, the answer is that different kinds of cells have what are called cell junctions that actually connect the cells together, sort of like two Lego bricks connecting together. Okay, so connective tissue is the only tissue that has cells and an extracellular matrix. Okay? And the purpose of connective tissue is to glue the body together, to sort of act as Elmer's glue for the body, where we need to hold body structures together. We need to bind them. We need to attach things to one another. That's the primary job of connective tissue. Okay. Purpose of connective tissue is to bind and hold structures together. Okay. Now, let's talk a little bit about this extracellular matrix and what it's made up of. Okay. So, composition. Well, okay. Cells. There's a variety of cells that are connective tissue cells, and we will talk about them later on. But let me talk about the extracellular matrix. Okay, the extracellular matrix is made of water and a combination of proteins and minerals. Okay, I'm going to use the word slurry, kind of like a slushy. It is a slurry of water, proteins, and minerals. Okay. And so the proteins that make up part of the extracellular matrix, well, the proteins would include substances like collagen, right? Collagen proteins, which form long chains that we call collagen fibers, they make up part of the extracellular matrix. And it's the proteins like collagen that provide a little bit of flexibility to structures like bone, okay? Can provide strength and flexibility to tissue. And then the minerals are primarily going to be calcium and phosphorus minerals. And these minerals provide hardness and rigidity to materials that need it. Okay. All right. So at any point, if you look on page 69 of your textbook, again, you see several examples of different kinds of connective tissue. And, you know, we'll be going in a deeper dive on some of this content in next week's lab time together. Okay. But for right now, let me point out a few things. Okay. When we talk about the types of connective tissue, I'll mention two broad varieties. If you look on page 69, you see along the left side column at the top, you have connective tissue proper. And that's going to include, let me do it this way. That's going to include what's called areolar connective tissue, which is important at various locations of the body, such as under your skin and so forth, and around your organs, and helps cushion the organs, helps provide protection to the organs so that when they push up into each other, they don't smash each other and damage each other. We have adipose tissue, which at this point, you know, adipose tissue is basically a biochemical battery, because adipose tissue, which you think adipose, you think fat tissue, fat cells. It's basically a whole bunch of cells that have triglyceride in it, and triglyceride is a lipid, and lipids are biochemically dense. You know, there's with energy. There's just a lot of energy in the chemical makeup of lipids. So let's see, this is used for energy storage in the form of triglyceride. All right. Here's a question for you, coming in your notes. Why is there a substantial layer of adipose tissue around your heart? Why is there a substantial layer of adipose tissue around your heart? Okay, Pete, to protect from damaging it. Okay, imagine that adipose tissue can provide some protection, although we usually think of areolar as being more of a cushioning capacity. Okay, but yes, it will protect the heart. And what else? Okay, let's see. Emily, why do you think there's a layer of adipose tissue around your heart?
So I don't know necessarily why. Trying to think of like an educated guess.
Okay, so Emily, you have to ask yourself, why am I asking you this question now? What did we just put into your notes?
Um, Connective tissue and I guess because it's a part of the organ system, you need different tissues to make up. And I don't know the exact answer.
Okay, thank you for trying. It's okay. And let me encourage you, folks, please, please be sure you are taking your own notes at home. Don't wait for me to give you my own notes because I don't want that back on the exam. What I want is evidence that you're processing the information. So Emily, we mentioned just briefly that adipose tissue is basically a biochemical battery. There's a lot of energy in the adipose tissue. So I'm going to ask you again, why is there a layer of adipose tissue around your heart?
Because your heart needs energy, I guess, to keep pumping.
Good, good, very good, exactly. Do any of us ever want to encounter a moment in life where our heart runs out of biochemical fuel to contract? That's not going to be a good outcome, right? So, you know, honestly, there are times when I have too much adipose on my behind. But having the adipose around your heart is generally a good thing because you want to have a biochemical battery able to fuel the heart muscle tissue to contract. Okay, let's keep going. Any questions right now? Okay, good job. Let's keep going. Okay. Mother Nature, we'll answer that question again and make sure it's in your notes. Okay, why is there a layer of adipose tissue around the heart? Mother Nature wanted to ensure that the heart muscle has easy access to a sustained energy source to power contraction. Okay. All right. Let's see. Okay, so we have areolar and adipose. We also have dense connective tissue proper. And for this one, I'll simply mention tendons and ligaments. Okay. You know that tendons are the type of connective tissue that attaches muscle to bone. Right? And if you know, if okay, I won't type it into the notes, but just a general question. Folks, what kind of injury is very common in the pitching staff of Major League Baseball? Okay, I can't deny that. I'm sure Major League Baseball pitchers get a lot of tendinitis, and they also get, you know, they need Tommy John surgery and all kinds of stuff. The one I'm trying to drive at this point is rotator cuff tears, right? You've probably heard of rotator cuff injuries, rotator cuff tears. Well, the rotator cuff refers to a group of tendons in the shoulder joint where if you tear any one of them, you may not even be able to lift your arm, okay? So tendons attach muscles to bone, and ligaments, well, ligaments attach bone to bone. Okay. Any questions, folks? Yeah, Jennifer, do you have are you said, Jennifer, you're asking what about the hamstring? Are you asking me about an injury to the hamstring? Is that what you're asking me? Right. Yeah. So, the hamstring muscle, the tendon of it is also a weak spot. It can also be damaged. Um, you might be thinking about the Achilles tendon in your lower leg, things like this. All of these tendons that are associated with these muscles, including the hamstring, are definitely weak points that get damaged during athletic training and athletic competition. Yes, the anterior cruciate ligament. I'd have to look that one up for you, Nick. Off the top of my head, I can't tell you about that, but I'd be happy to do that. Quinn, can a tendon wrap around a bone? Yes, unfortunately, it can. Tendons can wrap around bones. Tendons can twist over on themselves and create a lot of pain. I will admit that I recently experienced that when I was playing with my son outside. So yes, that can definitely happen. Okay, anything else? So do doctors untangle them? Generally not. Generally, given enough time and rest where you isolate the joint or the damaged area of the body will untangle them by themselves. So usually you don't need surgery for that kind of thing. But I can tell you through personal experience, it can be very painful. Okay, anything else? All right, let's see here. We need to finish connective tissue. So not only do we have connective tissue proper, but we also have specialized connective tissue. And I will list them out, but I'm not really going to describe them for you because specialized connective tissue is the topic of an essay question on your exam next week. So I'm going to expect you to do that work. Okay. So when we say specialized connective tissue, we're talking about cartilage. We're talking about bone. And we're talking about blood. Okay, so you will see on today's study guide coming out what I want you to do with that. Okay, so connective tissue made of cells plus extracellular matrix. And the function of this tissue is to glue body structures together. Anytime if you had epithelial tissue that needed to attach itself to muscle tissue, it would do so by having a layer of connective tissue in between. For example, you'd have epithelial, followed by connective, followed by muscle tissue. So the connective tissue acts as a glue or an intermediate layer of tissue between other tissues. And do you have any questions at this time? Okay, good stuff. So let's keep going here. Next tissue is going to be muscle tissue. Okay, so we'll go down here and start that. Muscle tissue is unique in the body because it's tissue that has the ability to convert biochemical energy in the form of ATP into mechanical motion. Okay. right? You contract your arm muscle, and the arm muscle that's connected to the bone via the tendons will move your arm, right? So that's the basic idea here. And that's only possible because the muscle tissue can break down the ATP that's in the muscle cells and use that to power muscle contraction. That's a topic that we are going to cover in considerable detail after your first exam in chapter 6, I believe. Okay. All right. So there are three basic types of muscle tissue, and they are skeletal muscle tissue, okay, skeletal, cardiac, and smooth muscle tissue. Let's take each one of these and describe them a little bit. Okay, so you have pictures of this on page 70, figure 4.3. Okay. All right, skeletal muscle tissue. What should we say about it? Well, we should say that skeletal muscle tissue is voluntary tissue. In other words, your brain, the conscious part of your brain can think a thought and your muscle will respond. Okay? So please do me a favor. What I want you to do right now is I want you to command your tongue to lift up and touch the roof of your mouth. Please raise your hand if your tongue obeyed your brain. Okay. So is your tongue a skeletal muscle tissue? It is, right? Your tongue is a skeletal muscle structure. Okay. All right. So skeletal muscle tissue is made of cells that are under voluntary, meaning conscious control. You can think it and the tissue will respond. Okay. Now, if you look very carefully at the pictures on page 70, you will also notice that skeletal muscle cells have multiple nuclei in them. And that's fairly unusual in the body, right? When we talked about cell anatomy in chapter 3, we defined the nucleus as the location where your DNA is, right? Where your genetic material is. And typically, we think about one cell having one nucleus. Well, skeletal muscle cells are peculiar in that they have multiple nuclei. Each cell contains multiple nuclei. That's a little weird when you compare it to the rest of the body. Okay, the reason that each skeletal muscle cell has multiple nuclei is that when your body was being formed and developed in your mother's womb before you were born, your current muscle cells that you have were themselves each made of multiple tinier cells. And as you are developing in the womb, these tinier primitive cells that each have a nucleus begin to fuse together like soap bubbles fusing in a kitchen sink. So tiny bubbles fuse together into one large bubble. And each of those tiny bubbles, each of those tiny cells fuses together, and they each contribute a single nuclei, so that the mature muscle cell you finally now have in your body, the mature skeletal muscle cell you have in your body, now has multiple nuclei. Okay. The skeletal muscle that we're talking about on page 70, if you look carefully, you notice that the picture in your book tells you that you have that the cells have these things called striations in it, and that striated appearance is very characteristic of skeletal muscle cells. If you look down, you also notice striations in the cardiac muscle, but they're even more prominent in the skeletal muscle. Okay. The striations are formed by a pattern of overlapping proteins that exist inside the muscle cells. Okay. Folks, how many muscles? Well, let me probably ask this at the wrong location. Let me ask it up here before we talk about types. Question. How many muscles are in the human body? How many muscles are in the human body? Okay, so I need to bring up the chat window here. Okay, do any of you know how many muscles are in the human body? Well, anybody? Right. Okay. Let me make this question a little bit more precise. I didn't do a good job with that. Let's put it down here. How many skeletal muscles? Right. Emily, you say 600. Jennifer, right, you had to look it up, and I appreciate that you did that. Okay. Right. 650 named. Folks, the answer is somewhere in the high 600s to low 700s. Okay. We cannot give you an exact number on that because different people that make their living as anatomists actually sometimes agree about whether one muscle is independent of another muscle. And so there's a little bit of disagreement among the professionals as to exactly how many independent, identifiable skeletal muscles there in fact are. But yes, I would say somewhere between the high 600s to the low 700s. Catherine, yeah, I'm a little suspicious of that 840, but you are certainly in the general ballpark there with your answer, okay? Okay, all right. And so, you know, you think about it, there are a lot of skeletal muscles that are required to allow you and I and every person to do the amazing movements that we enjoy doing, from simply standing up to doing a ballet, playing baseball, doing a martial art, whatever it is you enjoy doing. It requires somewhere near 700 skeletal muscles to allow that kind of exquisite motion. Okay. Let's go to cardiac muscle. Cardiac muscle. Okay, cardiac muscle is found in the walls of the heart. It makes up most of the heart mass. Makes up most of the heart mass. Okay. The cardiac muscle is involuntary, okay? That means you cannot consciously control your heart, okay? I won't stop sharing my screen, but as you are at home, please do this. Please order your heart with your brain to stop beating for the next 5 seconds. Fortunately for all of us, your heart ignored you, right? In other words, it did not pay attention to you. That's what it means to be involuntary. Okay, so cardiac muscle is involuntary. You cannot consciously control it. Okay. Cardiac muscle cells. Pete, but you can control your breathing to stop the heart. Pete, you can train yourself to control your breathing to influence your heart rhythm, yes, but you can't fully stop it, okay? Cells, cardiac cells are also striated, okay? And if you look carefully at your book, page 70, in the description of the cardiac muscle cell there's another phrase there that says specialized junctions. Cardiac muscle cells are connected to other cardiac muscle cells by very special junctions, which your book is giving a generic name to, and I'm going to put a more precise term on it. Cells are connected to each other by specialized junctions called intercalated discs. Intercalated discs. Okay. These intercalated discs, these specialized junctions, are made of groups of protein that allow an electrical signal to spread from one heart muscle cell to the next heart muscle cell in order to coordinate contraction of your heart muscle. Okay. Okay, it's made of specialized proteins that allow an electrical signal to spread between cells to coordinate the heart muscle contraction. Okay. And finally, moving on to smooth muscle tissue here, which is the third picture at the bottom of page 70, bottom of figure 4.3. Okay, the smooth muscle cells. Obviously, for skeletal muscle, you can pick any part on the surface of your body and find a skeletal muscle. And as we said, your tongue is a skeletal muscle. For cardiac muscle, well, it's obviously your heart, and you kind of know where your heart is in your chest, in your thoracic cavity. Okay, but for smooth muscle, it may not always be as obvious where we find smooth muscle. So smooth muscle is commonly found in the walls of your digestive system, and in the walls of blood vessels, and around organs that have smooth muscle tissue in them, okay? Around the walls of organs like the urinary bladder. We also have smooth muscle tissue around the hair follicles in our skin. So, you know, if you've ever had goosebumps because you were cold or scared, maybe sometimes the hair on your arm raises up. That's due to the contraction of small bands of smooth muscle tissue embedded in the dermal layer of your skin. Okay, so suffice it to say, there is actually a lot of smooth muscle tissue throughout your entire body. Okay, smooth muscle tissue is involuntary. Okay, if you don't believe me, then please with your brain order your carotid artery in your neck to close shut. Fortunately, it ignored you, right? Because if it didn't ignore you, I suspect you would pass out. So smooth muscle tissue is also involuntary. It just doesn't listen to you. Okay? And notice that smooth muscle tissue is not striated. Okay? Of the three muscle types we have, smooth muscle tissue is not striated. Okay. Folks, do you have any questions right now? Okay, let's finish with the fourth type of tissue, and that would be nervous tissue. Okay, nervous tissue is kind of interesting to talk about. Okay, so when we say nervous tissue, we're talking about the tissue that makes up your brain, spinal cord, and nerves. Okay, any tissue that's part of the brain, spinal cord, and nerves that come off the bottom of your brain or laterally off your spinal cord. That's all nervous tissue. Okay. Now, there's two major types of cells that make up nervous tissue. And those would be neurons and glial cells, sometimes called neuroglial or glial cells. Okay, for short, neuroglial cells and glial cells refer to the same thing. Okay, page 71 of your textbook, figure 4.4 has a microscope image that's been artificially colorized so that you can see a neuron and you can also see a neuroglial cell. They kind of look a little weird, but you know, that's what they are. Let me mention a few things about neurons and the glial cells. Okay. Neurons are probably the most well-known form of nervous tissue cell. I would be willing to bet that you've heard of neuron, and I'd be willing to bet that fewer of you heard of glial cells. Okay, so neurons are able to respond to the environment by creating electrical signals. Okay, so you know, if you're sitting at home and you have your window open and there's an odor of a skunk sitting outside your window, right? Obviously those skunk molecules are going to enter your nose. That's going to activate your olfactory nerve endings. We'll talk about that in chapters 7 and 8. And those nerve endings will send an electrical signal to your brain where neurons will help interpret what that signal is. And all of a sudden you have an image in your head of a skunk based on the odor. Okay? So neurons are able to respond to the environment by creating electrical signals. There are only two kinds of cell type in your body that are capable of creating electric signals. Okay, only neurons and a subset of cardiac muscle cells, which are not neurons, are capable of creating electrical signals in your body. Okay, only neurons and a subset of cardiac muscle cells, which are not neurons, are capable of creating electrical signals in your body. Okay, so what I'm trying to point out to you folks is that the ability of neurons to create electrical signals is a pretty unique property that most of the other cells of your body cannot do. Don't neurons use potassium and sodium to create the signal? Yes, they do, Quinn. And we're going to talk about that in chapter 7. Okay, so neurons have the ability to generate this electrical signal, which is pretty amazing. Okay, neurons have very, very limited reproductive capability, right? We kind of know this, right? Think about anybody that you know in your life that was unfortunate enough to suffer a stroke. And while hopefully that person that suffered a stroke was able to recover some of his or her lost capacity, it's also sadly often the case that certain capacity is not restored. Okay, and that's because neurons don't regenerate. They don't generate fast. They don't reproduce fast. And if they do reproduce, it takes them a long time. Okay, so neurons have a very limited reproductive ability, which is why a stroke can be so damaging. All right. We used to think that neurons couldn't reproduce at all, but now the scientific community has shied away from that. We do think they have some reproductive capability, but it's still very slow and very limited. And to be frank with you, if you have a stroke or if you have any kind of damage to the body of any kind, usually the impediment to repairing the body is the development of scar tissue. If you have a heart attack and you damage the heart muscle, scar tissue is going to develop. If you have a stroke and you develop scar tissue in the brain, anywhere scar tissue is present, it reduces the ability of your body to heal itself with functioning tissue to replace the job that was lost. Okay? And so one active area of biomedical research is trying to figure out ways to coax the body into changing the scar tissue into healthy functioning tissue. But we got a long way to go on that. Okay. All right. So that's the neuron. Okay. Neurons have 3 major components. 3 major parts to their anatomy. That would be the cell body. Okay, the cell body you see on page 71. Dendrites, which are short projections off the cell body, and axons. Okay, so we'll talk more about those at a later time, but your book's going to have them in there as you're studying this chapter. Now, the neuroglial cells. These cells, okay, let's see here. These cells cannot generate electrical signals, okay? These cells cannot generate electrical signals. Okay. And that's probably one of the reasons that for many years glial cells were known about, but they were kind of ignored because everybody was studying the neurons, because everybody wanted to understand the electrical properties of the neurons. And of course that's important, but you have to admit anytime you ignore something in the body, you're missing some important information. So neuroglial cells cannot generate electrical signals, so they're not as famous as the neurons. But the purpose of the glial cells is to sort of, at a very minimum, to be a caretaker, be a butler, be a nanny, be a nursemaid to the neurons. The job of the neuroglial cells is to take care of the neurons. They help support the health, functioning, and well-being of the neurons. So obviously, if they're not functioning properly, they can't do their job of helping the neurons function properly. So, you know, at least some neurological diseases may have more to do with dysfunction of the glial cells than they do with dysfunction of the neurons. One last thing I'd like to talk about a little bit is that the glial cells, while they cannot generate electrical signals, they can reproduce. They can reproduce like crazy, okay? So they have a robust reproductive capacity. And it's for this reason, it's the reproductive capacity of the neuroglial cells. It's for this reason that 99 times out of 100, if you have a patient that has a brain tumor, usually it's a tumor that's created from an overgrowth of the glial cells, not the neurons. Okay, so you might have known somebody in your life that's had a glioblastoma. A glioblastoma. Well, this is a brain tumor made of an overgrowth of glial cells, okay? Which explains why the large majority of brain tumors, like glioblastomas, are made of an overgrowth of glial cells. Okay. All right. Okay, gang. So, you know, this chapter is really a survey. We're introducing you to some of the features of epithelial, connective, muscle, and nervous tissue. But at various moments through our semester, you're going to get more specialized knowledge. For example, we are going to emphasize muscle contraction in Chapter 6. We are going to emphasize nervous system functioning and neurons and all that kind of good stuff, electrical signals in Chapters 7 and 8. So, you know, if it feels like this is somehow incomplete, you are correct, because there's more coming our way in future chapters. Do you have any questions right now? Okay. Well, folks, I have about 15 minutes with you, and so what I want to do is talk to you a little bit about, I guess I need to talk about cell junctions a little bit, and then we're going to talk about your skin a little bit. Okay, so let's talk about the cell junctions. That topic is on page 71 and 72. Okay. Molecules. Now let's put it this way. Structures that hold cells together. Okay. Cell junctions. Okay. We don't have to delve into this too much because your book only gives you a summary of it, but let's take a look at these things, okay? Again, if you have two epithelial cells, well, how do they stay connected to be part of a tissue? If you have two muscle cells, how do they stay connected to be part of a tissue, right? How do we connect connective cells to epithelial cells. So what are the structures that allow us to join cells together? There are more varieties than your book is telling you about, but the three that your book is going to introduce you to is a good starting point. Okay, first we have the tight junctions. Okay, the tight junctions, which you can visually see on figure 4.5. Okay, they're made of special proteins that interlock the plasma membranes of adjacent cells close to each other. They lock them together. Okay, so tight junctions are made of specialized proteins that attach and connect the plasma membranes of adjacent cells. Tight junctions, they form such a tight seal between neighboring cells that you can't even get water in between those two cells. Okay, they hold the cells together so tightly. Imagine that you take your two hands and you open your hands up and you smash the palms of your hands together like you're praying. Well, if you could push your hands together so tight that you couldn't get any water in between your hands, that's kind of similar to what a tight junction is like. So tight junctions form impermeable seals between cells. Okay, so tight junctions are going to be used in the body in areas where we need to control and regulate the motion of fluids, of liquids. Okay. So a good example would be your urinary bladder, where your urine is stored, right? Right now, the epithelial cells lining the inside of your bladder are joined to each other by giant networks of tight junctions. And that ensures that urine does not leak out of your bladder. Okay, how unpleasant would it be if our bladder leaked a bunch of urine out inside the abdominal space? That would not be pleasant. Well, fortunately, that does not happen because the lining of the bladder is made of epithelial cells that are liquid tight because of the tight junctions. Okay, so we have tight junctions as one category of structure that holds cells together. We next have adhesion junctions. Okay, adhesion junctions are different structures made of different proteins. Okay, you know, in science we know the names of many of these proteins, but I'm not going to give them to you because it just is not important right now. Okay, but this is a different kind of structure. It's also known as a desmosome. Okay, and what's important about adhesion junctions is that it's found in areas of the body that require a little bit of stretchiness. Okay. Okay, so just as an example here. So ladies, think about your uterus and the fact that, you know, if you're pregnant, there's going to be a baby growing in the uterus, right? Well, the structure of the uterus itself has got to be able to stretch to accommodate the growing child as the child gets bigger in size from month 3 to month 6 to month 8, right? It has to be able to stretch. So there's certainly adhesion junctions in between the cells that make up your uterus so that as the baby grows, the uterus can stretch and not tear itself apart. Okay. And finally, we have gap junctions. Okay, so gap junctions are a little bit of a different beast compared to the first two junctions we've talked about. You see a picture of them on page 72. And gap junctions are made of proteins that literally form little tunnels, little hollow passageways between neighboring cells. Okay. And so you're going to find gap junctions in areas of the body that need an electrical signal to propagate. So for example, you're going to find gap junctions a lot in the nervous tissue. You're going to find gap junctions a lot in your heart because of the electrical signal that has to propagate through the muscle anatomy of the heart in order to coordinate muscle contraction. Okay. So it's useful in structures that must allow the conduction of electrical signals, such as the nervous system and the heart. Okay, folks, do you have any questions about the cell junctions? Okay, I know we're approaching our end time here, and we probably all need a little bit of a break. Give me 5 more minutes, and then we will take our break before we start lab content here. Okay, so it's in this chapter 4 that your book introduces you to the 11 major organ systems of the body in figure 4.7. I think we answered that some time ago. Quinn, I see your question. How far can an adhesion junction stretch? I don't know off the top of my head, but if I had to estimate, I would estimate perhaps 200 to 500 nanometers. Maybe 500 billionths of a meter is what I'm thinking would probably be the right ballpark for that. Okay. Which obviously, 500 nanometers seems very, very small to us, but to a cell, that could be a huge distance. Okay. Okay, so back to the organ systems, right? We mentioned that there are 11 major organ systems in the body. Well, they are now defined for you on figure 4.7. Folks, don't try to memorize them. It's not important that you memorize them, but be aware of them, okay? So let's take what your book uses as the integumentary system and talk about it. Okay, so all right. So let's see here. An example of an organ system, okay? So your book uses skin as an example of an organ system, okay? So let's talk about that. We may not get this finished today, but it's okay, okay? So, yes, okay. So the integumentary system is the proper name of the organ system. Even though your book is talking about skin, it's more appropriate to think about the integumentary system. Now you might say to yourself, well, I don't like that because integumentary is a big, scary word. Don't worry about it. It's not that big of a deal, okay? The integumentary system is an organ system. Okay, this means it is made of multiple organs. Okay, Well, what are they? Well, here we go. Okay, the integumentary system is made of your skin, it's made of your hair, it's made of your nails, it's made of sweat glands and oil glands. Okay, so all of these structures are three-dimensional structures that have multiple tissues in them. Okay, so perhaps we should go way back up to the top. Now, hold on a sec. It would have been at the start of chapter 4. Okay, so chapter 4, we define an organ as a three-dimensional object made of multiple different tissues. An organ system is a group of organs that share tissues of a common biological origin. A group of organs that share tissues of a common biological origin. Okay. So, at the end of this chapter here, the organ system is the integumentary system. The integumentary system is the organ system. It's made of multiple organs. What are these organs? Skin, hair, nails, sweat glands, and oil glands. These all can be considered independent organs. Okay? Now, your book is going to focus specifically on skin as an example. Okay? All right. So, let's think about a component of the integumentary system, your skin. Okay. Okay. All right. So, have a look at figure 4.8. Okay, so if your skin is an organ, it should have multiple different tissues in it. Is that correct? If your skin is an organ, it should have different tissues in it. Is that a correct statement? Okay, Pete, thank you. Good, good. So let's take a look at figure 4.8 and show you how your skin contains multiple tissues. Okay, note the presence of multiple tissues that make up the structure of skin. Okay, all right. So just as an example, okay, just as an example, all right, first off, we have the epidermis. So the epidermis is made of sheets of epithelial cells laying on top of each other. Covering each other, okay? Made of sheets of epithelial cells covering each other. So there we have epithelial tissue, right? There we have epithelial. Next we have, let's take a look at connective, okay. This one it's a little bit harder to visualize it because it's everywhere, but it's not a discrete structure. So the dermis is primarily made of connective tissue, which means there are cells and extracellular matrix present. Okay, so you can see on page 75 the dermis. Folks, that dermis is made of cells. You know, some of the cells you can't see in that drawing because they're too tiny, but it's made of cells and material outside the cells, which is the extracellular matrix. Okay, so the dermis is made of connective tissue. And then we'll finish this topic and then we'll take our break. And then, let's see here. Take a look If you see figure 4.8 in your book, do you notice in the upper right corner there of the drawing, do you notice the erector pili muscle? The erector pili muscle and how it's connected to the hair follicle. Okay. All right, so this is connected to the hair follicle. It's part of giving you goose pimples and things, allows your hair to stand up. So here we have muscle tissue. The erector pili muscle is going to be specifically smooth muscle tissue. Okay, so we've got epithelial cells present, we've got connective cells present, we've got muscle cells present that are part of the skin. And to complete our picture here, you're going to find nerve endings embedded in the dermal layer. Along the left side of the picture, do you see the phrases sensory receptor and sensory nerve fiber? Okay, those are bundles of neurons, or really they would be bundles of axons of neurons that are embedded in the dermal tissue itself. Okay, so we have sensory receptors, which are made of bundles of nervous system axons, okay, that allow you to detect pressure on the surface of your skin. If somebody gives you a hug, you're going to detect that hug. You're going to feel the pressure of the hug by activating the sensory receptors. So here we have nervous system tissue, okay? So my purpose in doing this is simply to point out that the skin is an organ which is part of the integumentary organ system. The skin is an organ. It's a three-dimensional structure, right? You can look at your skin. You can see it. It has length, width, and depth, and it has mass. So it's an organ. And it's made of multiple tissues, and we've given you an example of all four tissues: epithelial, connective, muscle, and nervous. Folks, do you have any questions right now? Okay, I think we need a little break, so I'm gonna stop sharing. Folks, let's take a 15-minute break. Please be back online here with me at 2:20. And I'm thinking we might need 45-ish minutes, maybe an hour tops, to get through our micro content today, okay? So go take a little refreshment and be back at 2:20, please, and I will see you soon, okay?